Welcome to the Tic-Tective Podcast, a program of the Bay Area Lyme Foundation, where our mission is to make Lyme disease easy to diagnose and simple to cure. I'm your host, Dana Parrish, and I'm the co-author of the book Chronic, and I sit on the advisory board of Bay Area Lyme Foundation. This program offers insightful interviews with scientists, clinicians, patients, and other interesting people. We're a nonprofit based in Silicon Valley, and thanks to a generous grant that covers 100% of our overhead, all of your donations go directly to our research and prevention programs. For more information about Lyme disease, please visit us at bayarealyme.org. Dr. Mikki Tal is an immunoengineer, and she is the Associate Scientific Director at MIT's Center for Gynepathology Research. She's also the recipient of our Bay Area Lyme Emerging Leader Award. We are so proud of the work that she has gone on to do. She's been studying the effect of Lyme, COVID, and other chronic infections on the immune system with a focus on female health. I can't wait until you hear the discoveries that she's made. With no further ado, Dr. Mikki Tal. So you were the recipient of the Bay Area Lyme Emerging Leader Award. That's when I first met you several years ago, and I got very excited about your work. Was that your first foray into Lyme?
Absolutely, it was. That was the first time I met any patients and really encountered how devastating Lyme disease can be.
Yeah.
How underserved it is, how much of a problem this is,
Yeah.
and that really inspired a lot of my research going forward.
I'm curious how you got into studying Lyme where it relates to women's health, because that's been a big question mark and a big black hole, but a lot of us patients have had suspicions.
One of the things that happened is soon after that Emerging Leader Award, I was then doing all this research into Lyme, reading a lot about chronic Lyme and what was known and what was not. And I came across this review that had been written by a very prominent scientist in the field saying how you would be doing your patients a disservice to even consider chronic Lyme. One of the things that really struck me was that the sex differences that were being noticed in Lyme, that there were more diagnosed acute cases for males, and that there was more prevalence of chronic illness in females, that was being used to chalk this all up to hysteria, as opposed to really trying to find the biological differences that might point us to. And so I started thinking a lot about the sex hormones and just the role of sex in the response to infection. We know that all of our immune cells have receptors for these different sex hormones, but we hardly understand how that impacts their function. We really don't understand at a very fundamental level how sex differences drive differences in the response to infection. But we know that there are big differences. There's big differences in our responses to infection, in our responses to vaccination, so all sorts of immune stimuli. I started really doing a feminist revolution in my mouse room.
In your mouse room?
In the mouse room, exactly.
Okay, I don't think that many people get to say that. You have a mouse room.
Yes,
That's
a mouse room, a mouse house.
Oh my God, I love it.
Yeah. But so we had, the opportunity to really ask this question, to look at the infection in female mice, to compare that to the infection in male mice, to start to look at differences. And as we were doing that, I'll never forget this. There was this one particular endpoint experiment that, we needed to now, we have this incredible model and we try to get everything out of it that we possibly can. So we really try to use these experiments to ask every possible question that we could possibly ask. Everybody in my team knows, these kinds of endpoints are all hands on deck situation. Everybody has to help out. We're trying to harvest everything, look at everything. That kind of is a theme. So there's this endpoint experiment. There was this very chunky female, and we're joking about it a little bit. And I would later go on to feel so badly about that because when we opened her up, she was all uterus.
Really?
This enormous swollen uterus. And I'm talking about, we went down there with these little plates with these 24 little wells to put in these tiny little mouse uteruses. And this uterus was so big, and we needed like a 50 mil falcon tube to collect this uterus. This uterus was enormous.
So is this fibroids that you found?
This was not fibroids. So this is something that in animals would be called pyometra. So this is beyond just cysts. This is just tremendously swollen with a lot of liquid content inside.
What was it? If you could extrapolate to humans, if that's possible, what would it be diagnosed as?
I don't know. I'm not a gynecologist. I
Yeah.
don't know what this would be in humans, but what I do know is that when we opened up this uterus, there was a tremendous amount of Borrelia inside. It was just chock full of Borrelia. And so this thing was so horrifying to even think about, to just think about, from the whole process of it, just to think about, we were even joking, about how chunky this female mouse was. And
Yeah.
then that was all just this swollen uterus that must have been so painful. And it was so infected. And it's one of those moments where you look at something and you're like, nobody has ever looked at this before.
No.
Because you couldn't miss it. You couldn't possibly miss it. There's no chance, no matter what it was you had opened this mouse to collect, you couldn't have missed this.
Wow.
And so it just really dawned on us as a team at that point that nobody had looked. And, nobody had looked at this particular condition, this particular, length of infection, and then just looked at what was happening to the uterus. And so we look back at the literature and we saw, Janice Weiss had looked at Borrelia in pregnancy, 25 years earlier, and had reported that pregnant animals could get Borrelia infection in the uterus, but nobody had looked at this, in the context of just a female, a non-pregnant female. What would be the impact of a Borrelia infection on the uterus, on the vagina, on gynecological disease? And so, just as we started to frame that question, the more we looked, the more we found. And it was really shocking to me that nobody had looked, because at that point, it was very well understood that Borrelia colonizes the bladder.
That's a big thing you just said. So I had an early symptom of interstitial cystitis. I had it my whole life. I had it since I was little. I grew up in New Jersey, playing in the backyard, in the grass, in the trees. Definitely had Lyme, I'm sure. And when I treated it, my interstitial cystitis went away. When I treated it decades later, but I suffered with it my whole childhood. And so thank you for saying that, because I think that's a really important piece of information, because so many people are suffering with interstitial cystitis. It's torture, and they don't know, they're not told it has a potential cause that can be treated.
I mean, I think it's so critical that we really start to look at the data that we have access to in a new way.
Yeah.
After Lyme, what else then increases in risk?
Yeah.
And look for things that we didn't even think to ask about. But definitely from the bladder to the uterus, there's so much similarity that it begs the question. If the bladder is infected, we need to be asking if the uterus is also infected, and what's the impact on the vagina, and what's the impact on gynecological disease. These are, very close together, very similar. There's often an infection in one can impact the other. And so that felt very important. And I connected with Linda Griffith at MIT's Center for Gynepathology Research. And I asked her, actually, my question to her was, do you want to see something gross? And she said, always.
Yeah.
And when I showed her this female number 23, in our male versus female, her next comment was, okay, got to figure out how to bring you to MIT. And that's the beginning of how I got to the Center for Gynepathology Research, and for really just digging in deep into the sex differences in the immune response to infection and what is happening with gynepathologies with Lyme disease.
Could this have implications for endometriosis, for fibroids, for fertility problems?
Yes.
That is important. I know a lot of Lyme patients who had a lot of trouble conceiving and have a preponderance toward fibroids and endo.
We document that. So we collaborated with NASA Sina Armstrong's lab at Fred Hutch, and they did this incredible epidemiological analysis throughout multiple different data sets of American data sets, as well as we had collaborators looking in Finnish data sets. And across everywhere we look and we report in this paper, there's significant increases after Lyme in fibroids and in endometriosis. Just like your experience, when we put this paper online, we got just an overwhelming response from so many women who were like, you just told my story. This is me, and nobody had put this together before.
You're told, theoretically, if you're like me, you're going to ask your doctor, why would I have this? Oh, because some people are just estrogenic. So everything just gets blamed on estrogen. I'm not saying that some things are not estrogen, but I'm also not saying like maybe Lyme is impacting or these other hits. We know certain kinds of mycotoxins are estrogenic. Maybe Lyme is causing people to become more estrogenic. Is that possible?
That's something we are certainly hoping to find out. This is an active area of research for us.
Fantastic. Tell me about your work in Lyme in terms of how it evades the immune system.
So one thing that's known really well for Borrelia burgdorferi in general throughout the mammals is that if you don't successfully treat it with antibiotics, it will establish a chronic infection. And so there's no question or controversy about that. That is something that we understand very, very well. And in our mouse model, we take advantage of that. So in our mouse model, what we do is we infect the animals. We don't even bother to treat them. We just leave them infected and we let it establish a chronic infection. And so we have a mouse model for chronic Lyme, which is an untreated infection.
What's the window for treatment to, in your mind, eradicate versus go on to a chronic disease state, chronic infectious state?
So there's some really great researchers who have been really diving into this question. Monica Embers has done amazing work, really digging into the question of the effectiveness of different antibiotic treatments or combination treatments. In terms of timing, we know in the mouse model, it's pretty well established that if you would leave the infection untreated for over 2 months, the efficacy of antibiotic treatment goes down significantly. And so it becomes harder to treat at that point. And at that point, even if you treat the same way that you could have easily treated in the first few days, now maybe let's say 3 out of 5 of your mice would clear it, but not all of them, or something like that. This question wasn't about the efficacy of antibiotics, but rather using this model that we have, that we know Borrelia will establish a chronic infection if left untreated. And the whole reason we got into this question is that when I got that Bay Area Lyme Emerging Leader Award, I was a postdoc in Irv Weissman's lab looking at immune regulation and immune checkpoints. And so there was this huge boom in cancer research, incorporating immunology and immunological concepts to develop drugs like Keytruda and other checkpoint inhibitors that have really, really revolutionized cancer therapy. And the concept behind these checkpoint inhibitors is that the immune system has the power to kill you and therefore is very tightly regulated. And there's all sorts of brakes that you put on the immune system. And it's kind of stifled a little bit by these, there's checks and balances in place, and these checks really do hold the immune system in check. What they've done in cancer research is said, if we let up on this break, give you another shot, now can you clear these cancerous cells or this tumor we know you saw it, and we know you've been ignoring it because you're trying not to kill, because you get this window to just fully go into it. And if you can't clear it in that time, your immune system shifts into this place of, I see you, but I'm going to ignore this situation. The thing that gets really tricky with infection is that you have to know where you are in the infection to play with these checkpoints. So one of the things that we found is you play with these checkpoints early in the infection at your own peril, because the reason these breaks are in place, the reason there's these checkpoints, is that your immune system can kill you. And so if you play with these checkpoints and remove them in the early infection, the animal will just die. However, if you do it late, in these chronic stages, maybe you have a chance to now clear the infection. We did a lot looking at this particular checkpoint, CD47 SERP alpha, in the context of many different infections. And one thing that came up in my research is that the receptor, so what's driving the real break on that particular checkpoint, is extremely diverse in the population. So even you and I, ours might look different.
Yeah.
And I got really interested in what was driving that level of diversity.
Yeah.
And so likely it had been hypothesized that likely there was, pathogen pressure driving this diversity. So this means that we're constantly in this arms race between these invaders, and we're trying to get them out. And so they change and we change, and they change, we change. And so this diversity could have resulted from this kind of an arms race.
Yeah.
So what I thought is, in the Weissman lab, they had developed incredible tools to block this particular negative regulator. And I was like, why don't I just, use this to look? And Irv actually bet me about this and about what I was going to find. And he was right. So we ended up using one of these very, very potent proteins that we had made that could essentially act like this. Anything that this negative regulator could bind to, this protein would bind to.
Got it.
And so we were able to use that essentially to go fishing for any microbes that had evolved a way to mess with this negative regulator. And so when we did that, what we saw, so basically I walked around the halls of Stanford microbiology and I was asking everybody if they would test my protein, and we found a lot of interesting things. But one thing that popped up was Borrelia burgdorferi. And at the time, I was a viral immunologist. I thought bacteria were boring. And then this
You had not met Lyme.
Not Lyme. That's exactly what I realized. So Borrelia burgdorferi pops up, and I was like, I started reading about this bacteria, and I was, wow, this is no boring bacteria. This is really complicated. It's a complicated infection. It's a complicated public health crisis.
Yes.
I was hooked.
It's so common. That's the thing that also is not known by the research community at large. It's really, really common and causing so much different disease, like multisystemic, all kinds of weird stuff.
And I think that's getting more appreciated. I mean, it is the most prevalent vector-borne illness in the US. I think people are starting to realize that ticks are a real danger, and they're a real danger because of some of the really nasty microbes that they carry. And, one of them is Borrelia burgdorferi.
What is the role of microclots in Lyme and also long COVID or COVID?
Yeah. So I think to talk about that, we should talk about blood flow in general and lymph flow and just how things are normally supposed to move. So we're looking at a lot of different things in our clinical study, Maestro, and there's a lot of things that we know are going on in the infection-associated chronic illnesses, both in chronic Lyme and in long COVID. And so we're specifically looking for these things. And so, there's, I think, there's this incredible research that's been coming out of South Africa and other places really characterizing these microclots and what's happening with the immune response that's very, very sticky. One of the problems becomes a lot of things stem from having some ongoing insult. So let's say your barrier integrity is compromised, or let's say there's, and so now microbes are able to get in, or just things are able to get in that aren't supposed to, and the immune system's recognizing this kind of open wound.
Yeah.
Or let's say you have this ongoing infection that's causing this ongoing insult. And so the immune system is caught in the state of trying to respond. And one way that the immune system responds to things is through what we call a coagulation cascade. And basically a way of saying things get sticky. The reason things get sticky is problem number one that your immune system was made to address above all else and at all cost is an open wound, right? That's not tolerated. And so if there's an open wound, you've got to seal that wound. And you will do it at all costs. And so first you'll try really sticky stuff just to seal it up like you would maybe a flat tire. And then you'd even go to the extent of forming scar tissue there over what that tissue was over the function of the tissue. Anything to close a wound. You can't, you just won't tolerate keeping anything open. Normally your immune system is supposed to shift through these stages. The goal is not to kill you, obviously. That's not advantageous. Normally it starts off strong and then it'll exhaust and it'll ignore and it'll temper off. The problem is if you're stuck in this stage of an open wound, it can't shift. It can't shift because closing off is priority number one. And so all this sticky stuff that gets activated when there's some kind of opening and some kind of insult like that, as long as that is continually identified, you're continually making all this very sticky immune response and you're clotting things. And as we look at different diseases, we see that this has a lot of consequences. And so, obviously at the big level, we know that throughout your larger vessels, if you have a huge clot and you get a stroke and there's not blood going to an area of the brain, that's going to have huge consequences. But what if you get a tiny clot that is just blocking a little capillary? And now there's not blood and oxygenation getting to that area of the tissue. Right. What's the impact of that? A lot of people have this comorbidity of dysautonomia or POTS, right? So they're lying down and blood is getting to their brain, but then they try to stand up and blood is no longer getting to their brain. So the whole flow of the blood is off. We often don't think about all the things that have to go right just for you to be able to positionally get blood flowing to your brain against gravity. But you have to constrict these blood vessels and you have to change the pressure and you have to get this signal, immediately to get blood, to be forcing blood upwards to the brain as you change position. And if any of these things misfire, either the signals coming from the brain or the ability to constrict these blood vessels, because maybe the blood vessels are leaky or floppy, or maybe your blood volume is low, or maybe there's these clots that are impacting movement of the blood. And so there's all these things that can go wrong, but when any of them go wrong, you will absolutely feel that. And so that'll manifest as different issues that come with different symptoms. And so that's an aspect that we're studying in multiple different ways in Maestro.
Okay, so I am dying to talk to you about Maestro. The study started a couple of years ago. Tell me everything I need to know, and tell me how we can get into the study and who should be in your study and how we can rally the troops.
Okay, great. When we got to MIT, our mouse model was looking so interesting and there was a lot that we were learning from it, but we really, really, really wanted to start to ask these big questions in humans. And we were having a conversation actually with Emily Fairbairn.
The amazing Emily.
The amazing Emily. And talking to her about the study and what we could learn from it and what we want to ask And she was so excited about this and had so many ideas herself as well of, we could add in this and we could add in that. And from the patient perspective and from the place of, she has this saying of, turn pain into purpose.
Yes.
And so she has all these things that she's learned from herself, from her family,
Yep.
and just came to this with so many ideas. And we really kind of built together this idea of what we could study. And she made it possible to launch this study. So she donated this very generous gift to MIT specifically for launching Maestro. A lot of people in my team worked really, really hard to write these very in-depth surveys about really trying to ask about everything. And then there's this aspect of, you come in for the in-person visit, you get this suite of neurocognitive tests. So we can measure, we can look at eye tracking and electricity coming out of the brain, and we can look at dysautonomia. So we can have you lie down and then have you stand up. And in the meanwhile, you're wearing something in your ear, this Lumia device that's looking at directional blood flow. So that we can say, okay, when you're lying down, how much blood flow is there going through this? It's in your ear, but it's proximal to your brain. And there's a lot of correlation there between blood flow that's getting to your brain.
Yeah.
And we can look at what's happening there when you're lying down, when you're leaning. We can do capillaroscopy. So what I was talking about, looking at your capillaries for microclots, we can see into your capillaries by shining a microscope and a light right here, just under your nail, just at your nail bed. And looking here, we can see, are there hemorrhages in your capillaries? There's all these questions we can ask about the microvasculature. And what we can do is we can look at that before and after you do this test where you're lying down and then leaning. And so we've seen if you can make these necessary changes in your blood flow, and we're measuring in your ears, and then we're looking at it in your capillaries, and we're taking your blood pressure. And so then we ask our participants are incredible. And so until now, we've just added in these EV rings as a compensation, but until now, people were participating uncompensated. People who were sick were coming in. This visit could take them a good part of the day to get through. They were needing to rest. They were planning out, how they would come in person. They were planning out the, best possible conditions so they could make the trip.
And they come to you in Boston, right?
They come to us in Boston. And some of them know that, after this, they're going to feel even sicker, to help us document what's happening. And for me, the saddest thing has been the participants who tell us that they've lost hope for themselves, and they're participating because they hope we can figure this out to spare other people. That to me is so crushing, and it has to change. What I think MIT is really well positioned for is, what we want to do is we want to take these surveys that people are filling out. People know something's wrong, but that doesn't get them a diagnosis. Even if they get to a doctor who believes them, there's no diagnosis that comes from just these patient-reported outcomes. And so what we want to do is take all those things that people know about themselves, measure everything we can possibly measure, and then parse back, okay, this symptom, these are things you can measure. This is a number that we would see in healthy controls, in people who've recovered from COVID, who've recovered from Lyme. This is a number we see in people who didn't recover from COVID or from Lyme. And this is something you can track. And then this is now a basis for future clinical trials. This is a basis for writing textbooks for clinicians so that they know what the infection-associated chronic illnesses actually look like, and that they know what to measure. Because there's so many good-hearted doctors out there who went into medicine for the right reason. They don't know what to look for, and they don't have treatments. And this is a way to design clinical trials. This is a way to move forward.
So what do you still need? Who needs to show up and help complete this study?
So we really need acute Lyme cases, people who are within 4 weeks from their tick bite, ideally, or at least 2 months from their tick bite. And the reason we need acute Lyme so much, the reason this is so important, is so many doctors will ask us, how is all of this from Lyme.
Yeah.
And how can you know that it's all from Lyme? Like, so many things are going catastrophically wrong. Let's take this case scenario. This person got Lyme. They started losing their balance often. They fell. They got a concussion. Now they have all this brain fog. And the question becomes, is the brain fog from the concussion? Is the brain fog from Lyme? The power for us of having an acute Lyme cohort is that we can do all of these measures early in that infection before all of these other things, where we're really just looking at the context of this infection. And then we can connect with these people again 6 months later, a year later. How's it going? What's happened? Let's remeasure all these things. Let's track how they're going. And we see these numbers changing. We see things that are changing in accordance with how people are feeling. That is then so powerful because we can link it back to that original infection. You know, we can show that this person was working, they were, yeah, sure, maybe they got headaches and maybe they had some fatigue, but they were living their life and they were doing all the things. And then they get this infection and now pain goes through the roof. Fatigue goes through the roof. They're now not able to just live their life and do all the things. And you can tie it back to the infection. And so this is incredibly powerful. We are also comparing chronic Lyme and long COVID and healthy controls. And we are very carefully trying to match everybody. So sex matters, age matters. And so we do need people still to enroll, even though we've had an overwhelming response from the chronic Lyme community. And so thank you to everybody who has enrolled. We are going through these and very carefully trying to make sure we have matches across all of our cohorts so that our cohorts show, a similar range of sex and age across each of these different things that we're trying to compare.
So we need for acute Lyme patients, people who have a recent tick bite, do they have to have had a rash? Can they come to you with a bite without any symptoms? There are some strains of Lyme, in some cases, no rash is produced, but they still got Lyme.
If somebody has pulled off a tick that had blood in it and they sent that tick for testing and that tick was positive for Borrelia burgdorferi, they can enroll in our study. Right. So we know at that point they have been exposed for sure, that Borrelia has been deposited. And so that's sufficient to get in. If you got a rash, that's sufficient to get in. Right. If a doctor diagnosed you with Lyme disease, an acute infection of Lyme disease, that is sufficient to get in. If you got the antibody results. So there's multiple different ways to get in.
For people who don't know, because a lot of people still don't know this, if you get a tick bite and you get a rash, that is diagnostic for Lyme. It doesn't mean, listen, there's Rocky Mountain spotted fever. There are other kinds of rashes. So be aware that not every single rash related to a tick bite is Lyme, but most of them are. Many of them are. And they don't look like a typical bullseye often. They can be really light pink, may not have a clearing. I've tweeted out my picture of my Lyme rash, which was extremely uneventful looking and very low key right on my shoulder. So I'm just pointing this out because I want people to get to you quickly and to recognize the signs, because that's your diagnosis. When you see that rash, that is Lyme. There is no early blood test for Lyme that exists.
Not everybody gets the rash, but if you get the rash, consider yourself a lucky one, because that is an early diagnostic and that means it's time to treat.
Absolutely agree. Yeah, great point. So we didn't talk about the connective tissue stuff, but I want to talk about that because I also heard that you had a fabulous exhibition last night and you got to spill the beans, girl.
All right. So last night we invited people to come out to MIT, take a look at some of these incredible microscopy images, take a look at some of the things that we're doing in Maestro. And it was really incredible. We had a mix of scientists and engineers and artists and people who are interested in Lyme coming out to look and really getting to kind of see the unseen in a way that I'm so excited about sharing this with people. I think it's really important for people to see. I mentioned that I was very moved at that event where I got the Bay Area Lyme Emerging Leader Award. Yeah. I met there this incredible person who's been through it. I think Nicole.
Nicole Malachowski, and I'm just a huge fan. I'm like her
Incredible.
fangirl. I love, she's the first female fighter pilot. She talk about a superstar.
So she told me that she needed to see her enemy. I thought about that a lot, and I thought about what that could mean for people. And when we started getting these incredible microscopy images through, being at MIT, we can collaborate, especially in the School of Engineering, we can collaborate with labs who are developing microscopes unlike anything you've ever seen, seeing deep into the tissue.
Wow.
And so we have this incredible collaboration with Sishenyu. And suddenly we can see the Borrelia, and we can see the Borrelia where they are. And we can see them nestled in the collagen, in the connective tissue. And so seeing this, I was thinking other people have to see this. And I put some of the early pictures on Twitter and Blue Sky and LinkedIn, and people responded to me saying things like, to be able to visualize this thing I could never possibly see with my eyes, I just started to think about the power. So that was really special. And what we were actually showing people is that the Borrelia is nestled right up there in the collagen. And what's happening is that we show that in some mice, the immune system is responding in a way that's protective. It's checking it out, but not causing a lot of damage. It's there, it notices, but it's, mostly leaving the Borrelia alone and leaving the collagen alone. In a different strain of mice that gets more allergic-y, they're sending mast cells to the Borrelia to attack, and they're damaging the collagen.
There you go.
Because the mast cells are going for the Borrelia that's in the collagen, and the collagen's paying the price.
We always say it's the immune response to the infection. I mean, we say it so often because people think that their bodies are teeming with these microbes, but that is not necessarily the case. Could be a small amount, and you get this huge response, right?
That's right. That's right. And so I think, when I think about immune dysfunction in this context, I'm thinking about immune dysfunction on many levels. One, immune dysfunction is just not being able to clear an infection. But as we talked about earlier, Borrelia establishes chronic infection that mammals can't clear. Yeah. So this is an inherent immune dysfunction that this bacteria has just found a way to evade our defenses. But then another immune dysfunction is responding in a way that is very harmful and that has a lot of collateral damage, and that will remodel the tissue in a way that it's going to really hurt and is going to cost you. This manuscript that we're working on right now, and just really diving deep into what's happening at the connective tissue. And this is, really spearheaded also by a research scientist in my lab, Beth Pollock, who came in with these big questions about the connective tissue, and is the first one that really even got us to ask this question and to look at what's happening here in terms of the damage to the connective tissue.
Could Borrelia be causing Ehlers-Danlos? We talked about interstitial cystitis. That's been known for a really long time. There's always this big push to peg everything on genetics, and that's just simply not true. And in the case of Ehlers-Danlos, that's a big hot button topic. So, do you think that these infections could be causing some cases of Ehlers-Danlos?
It's a huge hot button topic. And this is another place where our mouse model becomes extremely powerful. And I know that right now in the current climate, everybody's like, move over mice, do everything in human. And the thing is that mice have also been evolving with Borrelia burgdorferi for a very, very, very long time. And in mice, we can still watch this infection play out in a natural host that can't clear this infection and that has different genetics and different immune responses. With mice as well, we can play out a lot of the EDS genetics. So for the types of EDS where we know which gene, we can make those mice, we can see that connective tissue damage, we can see the impact of it, we can get what happens in EDS is in our EDS mouse models, you can tie a knot in the tail. So the tail rigidity is so hypermobile and it's so lax that you can just tie a knot. And you should never be able to do this in a mouse tail. It should be so rigid that this wouldn't be possible. But this is possible in the EDS models. In our uninfected mouse models, you cannot do that. They are not an EDS model. They do not have the genes that we know drive EDS currently. However, we found a strain that if you infect it, it will develop hypermobility, and you will be able to tie the tail in a knot, like an EDS mouse, but it is not an EDS mouse.
Do you differentiate based on just the genes? So what you would feel you can call EDS versus what looks exactly like EDS but isn't called it because they don't have that genetic marker?
We collaborated on a study from Mayo Clinic asking people with long COVID, all these different survey questions. And one of the things that popped out there was the significant increase in hypermobility spectrum disorder and EDS, significantly increased, and even more so in females. Looking at that in Lyme, I think is so important. In the mice, can we say if this is EDS or hypermobility spectrum disorder when they don't have the genes and this is not genetic, and we have forced this to happen through letting an infection go untamed? No, we can't say that. But what's really, really powerful about this mouse model is that we can say that this happens, this hypermobility happens. In the future, we can look and say, is that EDS? Is that not EDS? But we can say there's this infection, and when you don't have the infection, the tail is not hypermobile, and the spine, and we're looking at the integrity of the space between the vertebra, throughout the body. And then what we're able to show is that if in this particular strain of animals, of mice, if you infect them, now this will fall apart. And it wouldn't have fallen apart over that same amount of time, that same age, had you not infected them. The power there is, comparing infected and uninfected, comparing an infected animal of this strain versus that strain. So the strain that makes a really great immune response that's protective and not harmful, this isn't happening. Now we get to look at all these comparisons and really try to understand from a very fundamental mechanistic level what's happening. I think names will come later, and there's a lot to clean up in names across. And now what we're doing is we're working from the bottom, working our way up piece by piece and saying, okay, this happens, then that happens. And let's describe it mechanistically.
I actually didn't know that EDS was such a hot button topic, and that there's a group of people who think it's only genetic, because coming from the Lyme world, it's known that Lyme can cause it. Or it's really thought, and it's now been Ed Breitschwart and I talked about it. I think he published on it for Bartonella as well. So we know that these bacterium have an affinity for collagen. They attack collagen. They eat it, however you want to think about it. And it just stands to reason that people need to be open-minded about this, because if you want to, not everybody has to live with this. You can treat it in these cases where infection is causing it. At least I've seen many, many cases get treated. I've seen these conditions come to a halt. I've even seen some of this reverse, like in my case with interstitial cystitis. So I just feel like there is no reason to be dogmatic about this, because learning about it and understanding that it could have an infectious cause that can be treated is like hopeful and amazing.
Oh, absolutely.
So I don't really get the it's only, I've talked about it a couple of times publicly, and people like flip out. I'm like, I'm shocked by it, and I don't get it at all, because I wanted answers my whole life. When I finally got them, like my mind was blown, and then I had to go write a book about it with Dr. Phillips. So it's like, that's how I've got to feel passionate like you do about this.
And I think that, one of the important things is, does it even matter in terms of the chicken and egg? So yes, maybe there was an underlying vulnerability. Maybe there was already something about the collagen that was a little weak, or there was some risk there that, had it not been aggravated, you would have never, it wouldn't have bothered you in the same way, but was there a genetic underpinning? Maybe.
It doesn't, but does it matter? That's my question. To me, it doesn't.
Exactly.
There's this concept of epigenetics, and people don't take into account that you may have a predisposition to something. You get this hit, as we call it, and then it expresses itself in that way. And it's just an important concept.
Agreed.
I'm so curious how you navigate the chronic Lyme dogma. So when you encounter clinicians or researchers that say chronic Lyme is fake or long COVID is fake, these are psychiatric diseases, psychological, psychogenic diseases, what do you say? And have you ever converted a skeptic?
So this is an interesting one for me, because despite the fact that I'd done my PhD in Connecticut, and I lay it out in the grass, in the sun, and I never thought about Lyme. As I said, I was a viral immunologist. I mean, I knew about Lyme, but I didn't really think about it. And it wasn't until Borrelia burgdorferi popped up in the screen in my postdoc years later that I really started to think about it and to think about what people had been through. And when I realized this whole thing about the sex differences, that was the first time that I realized that the fact that I was a woman can and will be used against me in medicine to discriminate against me if I get sick. I was horrified by that. And I realized that we didn't have enough women asking the right questions in places of power that were making funding decisions. That's when I really started to feel like I needed to be here. Not as somebody who's ever had experience with Lyme or even family with Lyme. Like I'm not coming into this from that place, but intersected into it from the place of from being a woman, from just seeing what I'm seeing. And what I started to realize is it's hard when you're a doctor and you're seeing patients quickly and you are seeing patients that you don't know how to help. You don't actually know what's wrong with them and you don't know how to help them. And I think that some people have fallen into the trap of this is psychosomatic, this is in your head. And that's a nice, easy way to reassure yourself that you didn't fail this patient. The patient has some issues.
Right. It's the patient.
Yeah, I actually had a situation that I was introducing myself to a neighbor and they asked me what I work, what I do. And I said, I work on chronic Lyme. And they said, that isn't real, right?
Oh my God.
Yeah.
I love that they think they're telling you something.
I know. And I was like, oh, wow. Thank you for, informing me about this. And
thank God they let you know before you went to, spent any more money on research for this fake disease.
What's neat is that because I have this person as a neighbor, I have used them as my skeptic benchmark. And so as I get
medical field by chance, or are they just regular civilian, medical? So MD, or I'm just curious, like
this is an MD. And so I have used them as my benchmark as I have more research to see what convinces them, and I have actually brought this person to the point that they said, okay, tell me what to do, then I've been doing this wrong. Tell me how to do it differently. Now we're not there yet, but I feel very proud that I have at least gotten to this point of convincing evidence that this is not hysteria. This is not women seeking, to explain away menopause and regular aches and pains. This is a horrible disease. We have left people to rot, really just without access to our normal. I mean, we always talk about the cracks in the medical system, but this is a chasm. Yeah. And I really do think that maybe one silver lining of COVID, and I hope that this is true, is that because long COVID happened to so many doctors, so many nurses who had the medical vocabulary to describe what was happening to them, whose colleagues knew them from before, that it became impossible to ignore what was happening. And I think this may start to really be trickling to giving a voice to ME/CFS and chronic Lyme and all these other infection-associated chronic illnesses that never got a name. Long flu never even got a name. And I hope that this really is changing.
That's an incredible story, by the way. I have gotten a number of emails or messages from doctors and clinicians who were non-believers who became converted after hearing something that I said or seeing an interview that I did with somebody like you. And it's extremely gratifying. It's like, this is why I keep at it, because it's going to trickle down to their patients or to their research or to conversations with their colleagues. And that's the hope. It's not going to be taught in med schools, I don't think, anytime very soon. Maybe I'm wrong. Hopefully I'm wrong. But till that day, they need to hear from people like you. Before we go, what are you most excited about in terms of the future of diagnostics and any other research that maybe we didn't touch on today?
What I'm most excited about and hopeful for is that we enter this new era where we have a more foundational understanding that allows us to really treat intelligently. And so, if people are becoming allergic, that we can treat that, or if they really have an ongoing persistent infection, that we can treat that, but where we can really start to parse these things out. And the other thing I'm really excited about, every day I'm excited to be doing this research at MIT, because I feel like it's really, really special when you're in a place that has this incredible technology know-how, these engineers that just know how to build stuff that works, and they hear about Lyme and they're just like, wow, that's a big problem. I want to help, and just, I really feel like I'm in the right place to do this research and for things to really change.
Thank you for joining us for this episode of Tictective, a program of the Bay Area Lyme Foundation. For more information or to get involved, please visit us at bayarealyme.org.